Okay, good afternoon. Can you hear me? Anyhow, good afternoon. Good to see everyone here today. We've been planning this meeting for quite a while. My name is Joel Walensky, and I will be the host for today's seminar. And I'd like to make a few quick announcements as we begin today. I wish to thank, first thing I'd like to thank Livingstone Fellowship and Share the Power for sponsoring today's seminar, and also for Livingstone Fellowship for providing us for the facility here today. So would you please give them a round of applause for that? Thank you. Now the other thing I'd like to announce that we are live streaming the event today, and we will be recording it. So I just thought I'd let you know that. And today's seminar is going to be in two parts. So we'll have a break between the two parts, so you won't have to sit here too long. So we'll give you a break. So each part will be hopefully under an hour, approximately under an hour. So we will have a break, between the two. So I just thought I'd let you know that ahead of time. And also we have a table at the back of the church there. I haven't seen too many people go there yet, but we'd like you to sign up with your email address if you're interested in finding out about future events that we may have relative to this subject and similar topics. So we're anticipating in the future doing some other things, because I know I personally feel that this subject is very, very important, and you'll see as we go on. So if you want, please go to the back table and just give us your email address. I would appreciate that. Okay. I'd like to provide a quick background to how this meeting happened today. Okay, and it goes back a little ways. About 29 years ago, I myself received a magazine that had on the front cover a drawing of Dr. Behe and Charles Darwin. Would you put the slide up, Roger? And there's the magazine that I received in my mailbox 29 years ago. And there's Dr. Behe on the left, and he's playing poker with Charles Darwin. I must admit I had never known either one of them before. So that magazine, that cover is from 1997, okay? And below that is the topic. It said, Meeting Darwin's Wager. And that was the topic in that magazine. There's a very lengthy article about what we're talking about here today, okay? Like I say, that was the first time that I had ever known about this subject myself. And I was also surprised as I read that article 29 years ago to find that Dr. Behe lived in the Lehigh Valley. I said, that's amazing. I was really surprised by that. Now, in that article, the first sentence of the article reads as follows. If you go to that magazine, I actually have it in the back if you want to see it. This is the very first sentence. During the fall of 1996, a series of cultural earthquakes shook the secular world with the publication of a revolutionary new book, Michael Behe's Darwin's Black Box, The Biochemical Challenge to Evolution. Will you put the next slide up, Roger? And there's the book in its first edition from 30 years ago. Later on in the article, there's a quote by a philosopher, David Berlinski. And remember, this is from 29 years ago, okay? David Berlinski had this quote in the article. Darwin's Black Box is simply an extraordinary piece of work that will come to be regarded as one of the most important books ever written about Darwinian theory. No one in the evolutionary camp can propose to defend Darwin without meeting the challenges that Behe has set out in his book. It's really quite compelling. Those are the words written 29 years ago when the book was first published. Also in the same issue of that magazine, Dr. Behe's book received the magazine's award of the 1997 Book of the Year. So Christianity Today awarded that book the Book of the Year less than a year after it was published. That was almost 30 years ago. Now, so when I got that magazine, that's essentially what woke me up to this subject. And it's been a journey ever since. Now, about 10 years later, my daughter came to me one day and said, "Dad, you might be interested. Dr. Behe will be speaking at our high school next week." I said, "Of course I'm interested," and I went, okay? And that was the first time I saw him in person. A little later, I found out that Westminster Seminary in Philadelphia sponsored a conference on science and faith in Philadelphia, and I attended that conference for over 10 years. I think it lasted about 12 years or so, that every year they had a very high-level conference on intelligent design. Dr. Behe spoke several times. Like I say, I know I attended it myself, 10 times, 10 years. And so that was, part of my journey in this subject. But then about a year or two ago, I found out that they decided to discontinue the conferences, that they weren't going to be putting them on anymore. So it went for, like I say, for quite a while. And I have some of the materials back there from that conference at the table. And so that's what gave me the incentive to start thinking about doing something locally in the Lehigh Valley in Michael Behe's backyard, since these conferences wouldn't be in Philadelphia anymore. So I pursued that, and here we are today. And I was so glad that Dr. Behe agreed to come. Let me give just a few words about Dr. Behe if you don't know him or who he is. He was born, raised, and went to school in Pennsylvania, and then moved away from Pennsylvania for a few years. And then last year, he celebrated his 40th anniversary as a professor at Lehigh University. So he's been with us for 40 years here in the Lehigh Valley, which is pretty amazing, right? But this very year, in a few months, it will be the 30th anniversary for the publication of his book, Darwin's Black Box. So it's been here for 30 years, and so it's quite amazing. Today's presentation will be divided into two parts. The first part will be the basics of Darwin's Black Box, in case you're not familiar with the book or know what it talks about, and so you'll get a nice introduction to the book. And then in part two, we're going to have Dr. Behe talk about what's happened in the last 30 years in this field. And a lot has happened, okay? His book was sort of really the explosive start of everything in this field. Also, we will not be having a formal question and answer session today, but I encourage you, if you have questions for him, come up to him, talk to him during the break or after the meeting or whatever. If you have something really stirring that you'd like to ask him, just talk to him personally. He's more than willing to do that. And I myself consider Dr. Behe to be one of the most well-known people in the intelligent design field. He's not the only one. There's a multitude of people that have written on this subject, and I have a selection of books in the back there, you see, by various authors, mostly very scholarly books. It's not just some, black magic thing at all, okay? But I believe he's most well-known because of the publication of that book. When it came out, it was ready for that book. And so he was the one who stepped up and brought it to us. So with that today, I would like to invite Dr. Behe to come up to the stage. Would you give him a round of applause as he's coming today? Thank you very much. Okay. At this point, we'll turn it over to him. Thank you.
Thanks very much. Very much, Pastor Marcus. for having me here. And as Joel mentioned, there is going to be live streamed. And I don't know, can you see, there's a kiss cam over in the corner, so just keep that in mind. Okay, so the title of my two talks for today is going to be the argument for intelligent design in biology. And it's going to be why it is rationally compelling, based on the physical, biological evidence, to conclude that life has been designed. And the argument's going to have 5 sections. And the first section is that design is not mystical. It's not something you have to, close your eyes and, touch your head to see. You deduce it from the physical structure of a system. The second point is that everyone agrees that many aspects of biology appear to be designed from that physical structure. And when I say everyone, I mean even those folks who strongly disagree with the ultimate conclusion of design. Rather, a lot of people think that Darwinian evolutionary processes can explain the appearance of design. And so the third point is going to be that there are structural obstacles to Darwinian evolution. There are physical reasons to think that it cannot do what its proponents claim for it. But if you're like me and you open a magazine or turn on a nature show, you are told that science already knows that Darwinian processes can explain all of life. And so the fourth point is going to show you that those grand Darwinian claims, not little Darwinian claims, but those grand Darwinian claims rest on undisciplined imagination. And imagination can be good, but it can be a two-edged sword on occasion if it's undisciplined. And the final point then is going to be a kind of a summation point that right now, in the year 2026, we have strong evidence for design in hand, and yet little to negative evidence that Darwinism can do what is claimed for it. So I'm going to go through these points in the first talk in order. So let's begin with that first one, that design is not mystical, rather it's deduced from the physical structure of a system. But before we do that, we have to decide what is design. And its discussion goes back a fair amount, a fair number of years, but a large figure in this field is this fellow here, a philosopher named Thomas Reid, who was a Scotsman, and wrote in the 18th century an essay on the intellectual powers of man. And he made the observation that the general principle that, the design principle, the general principle that intelligence in the cause can be inferred from its marks or signs in the effect. We cannot read minds. So it turns out that the only way we apprehend whether someone is intelligent or whether something that was done by an intelligence is by what was done, its marks or signs in the effects. Well, what marks, what signs? Well, if you go to the dictionary and you ask yourself, what does design mean? There's a lot of senses of the word, but the relevant one is the following, that design is the purposeful or inventive arrangement of parts or details. And that's a little wordy, but we can shorten it and say design is the purposeful arrangement of parts. That's the outward effect of intelligence, the purposeful arrangement of parts. And that means that we infer design whenever we see that parts appear to have been arranged for a purpose. And that's a bunch of words, but oftentimes it can help to see an example of it. So let's take a look at this next slide here. Now, this is a Far Side cartoon. And as you can see here, we've got a troop of jungle explorers, and the lead explorer has been strung up and skewered. And this guy here turns to this guy here and says, "That's why I never walk in front." Words to live by, I can assure you. Now, everybody here in this room looks at this cartoon, and you immediately know that this was designed. That was not an accident. As a matter of fact, the humor of the cartoon depends upon you recognizing the design. It wouldn't be all that funny if he had just fallen over a cliff. So how do you recognize that design? Is it a religious conclusion? Probably not. Now, you recognize the design because you see a number of parts that have been arranged for a purpose, the purposeful arrangement of parts. We recognize design from the physical structure of a system. But there's one other point here that you have to keep in mind, otherwise you can get confused and easily kind of sidetracked. And that is that the strength of the inference is quantitative. And that just means that the more parts you have and the more closely they fit the purpose of the system, then the more and more and more confident you can be in the conclusion of design. And again, that's it's good to have an example of that. So let's look at the next slide here. Suppose you and a friend were walking along in Idaho and you came by these mountains, and he asked you, "How do you think these mountains got here?" You might say, "Well, I never did take that geology course. Maybe plate tectonics or volcanic activity." Okay. Probably most people would be happy with that. But suppose you and your friend took an even further walk and you came by this mountain. This is called Old Man of the Mountain in New Hampshire. And he asked, "Okay, well, how do you think this mountain got here?" And you might say to yourself, "Well, gee whiz, this looks like it might be a forehead. This maybe a nose, and a mouth, and a chin. Do you think maybe some prehistoric tribe might have..." Nah, that's silly. No, it's just a funny arrangement of the rocks. And this actually fell down a number of years ago. These are government workers here trying to shore it up. And if you know anything about government workers, you realize it would fall down later on. But nobody here works for the government, do you? Well, suppose you and your friend continued your walk and you came by these mountains here. And he asked you again, "How do you think this mountain came to be?" You'd never say plate tectonics or volcanic activity. You'd immediately recognize that these were purposely shaped. They were made to resemble presidents of the United States. And you can tell that because you see the way that all of the parts, the shapes of the rocks, are fit for the purpose of portraying those images. So the point is that you can have more and more and more evidence for design. And the more you have, you can quickly arrive at absolute certainty that something was designed. So it turns out that a purposeful arrangement of parts is the way, the only way that we recognize the work of a mind. Whether it's the shapes of stone to make it look like a face, or whether it's sounds, the arrangement of sounds to produce words in a conversation, or strokes of a pen to produce writing in letters and books, or whether it's events in a plan to carry out some goal. The way that we recognize the work of a mind is in the purposeful arrangement of parts. Now, what kind of an argument is that? What's the structure of this? When we see that whenever we have something like a purposeful arrangement of parts, whenever we see something that we always realize, or we always find, that is produced by a certain cause, or it leads to a certain conclusion. What kind of a structure is that to the argument? Well, it's illustrated on the next slide here. Who can tell me what that is? A duck. Good. And how do you know that? Yeah, if it looks like a duck, and if it walks like a duck, and it quacks like a duck, then it's rationally compelling to decide that this is a duck. So this is actually the colloquial way to look at this type of an argument. But philosophers know that this is actually one of the two major ways that we do make arguments or come to conclusions. And as a matter of fact, they have a technical name for it. They call it an inductive argument. Hey, they thought of that word. I didn't. Okay, inductive argument, philosophers. So are we talking philosophy or what? Well, no, if you look up inductive reasoning in a reference work, you find something like this. Inductive reasoning. When a person uses a number of established facts to draw a general conclusion, he uses inductive reasoning. This is the kind of logic normally used in the sciences. So an argument for design based on a purposeful arrangement of parts is a scientific argument based on empirical physical evidence and the normal modes of reasoning that we always use. Okay, so that was the first point, that design is not something mystical, that we deduce it from the physical structure of a system. Let's go to the second point now, that everyone agrees that aspects of biology appear to be designed, beginning from throughout history all the way up to the present, even folks who disagree with the conclusion. So let's look into this a little bit and see at the very beginning, how has scientific knowledge affected our apprehension of design in life? Let's go back to a time when science was very primitive or non-existent. Here's a painting of King David, and in Psalm 139, as you know, he wrote, "I will praise thee, for I am fearfully and wonderfully made." Here's a question. How did he know he was fearfully and wonderfully made? Is it because God told him, "You're fearfully and wonderfully made"? Why didn't he think it was kind of shoddily made or kind of a glob or not all? Well, because he could tell. He could see, holy moly, look, I got these fingers. I can play the harp. I've got this strong arm. I can shoot a bow. I've got eyes. I can see great distances. He could tell. It wasn't because somebody had to convince him. He could tell that he was fearfully and wonderfully made. Well, the ancient Hebrews, of course, were many things, but one thing they weren't were sophisticated scientists. And for a long time, it took time for science to develop. And 1,000 years later, folks in the classical era had started to learn about anatomy and dissect animals and even some humans and so on. What did they think about what they learned once they could see things more systematically than people before them. Well, one of the leading scientific lights of the classical era was this guy named Galen, who was a Roman physician, and he studied anatomy, and he wrote that the human body is the result of a supremely intelligent and powerful divine craftsman, the result of intelligent design. And the point is that the more he knew about life, the more he was impressed with the design of life. And this trend continues. In the Middle Ages, in the 1600s, there's this guy, Anton von Leeuwenhoek. He was one of the early microscopists when microscopes were first examined, and he was one of the first people to see single-celled creatures and tiny organisms in swamp water and so on. And he just exclaimed over their design, too. He saw how well structured they were for their way of life. This continued even up to the year 1800. A man named William Paley, who was an Anglican clergyman at the time and also a good scientist, as clergymen oftentimes studied science back in those days. And he wrote a famous book called Natural Theology, which opens with something called the watchmaker argument, which says that if you found a watch in a field, you stumbled over it, you picked it up, and if you looked to see how the watch worked and how its gears and parts were arranged to tell time, you would immediately know that it was designed. So a purposeful arrangement of parts. And he went on to argue that what we know about life, and the universe at large too, shows that it is even more purposefully arranged than a watch. And so we have even more reason to think that living things are purposely arranged, have been designed. But about 50 or so years after Paley, Charles Darwin wrote his book, and science has accumulated knowledge with increasing speed. So what do people now think about whether life looks designed? Well, it turns out nobody has changed their mind. As a matter of fact, in 1986, 40 years ago, Richard Dawkins, of course a famous atheist, wrote in a book he called The Blind Watchmaker, kind of a takeoff on Paley's argument. On the very first page of the book where he strongly defended Darwin's argument, he writes, "Biology is the study of complicated things that give the appearance of having been designed for a purpose." That's the very definition of biology, according to Richard Dawkins, things that appear to be designed for a purpose. And, why do they appear to be designed for a purpose, according to Dawkins? Is it for aesthetic reasons? Maybe because flowers are so pretty and baby seals are so cute. Is it for aesthetic reasons that they look like they were designed? Well, no, not according to Dawkins. According to Dawkins, life appears to be designed for engineering reasons. He wrote, "We may say that a living body or organ is well designed if it has attributes that an intelligent and knowledgeable engineer might have built into it in order to achieve some sensible purpose, such as flying, swimming, seeing. Anybody can recognize an object that has been designed just by looking at the structure of the object." So he agrees that's how we come to a conclusion of design, when we apprehend a purposeful arrangement of parts. Of course, Richard Dawkins thinks that he has another explanation for this, but he agrees that biology looks like it was designed. Well, we said that design, can be greater and greater and greater depending on the number of parts and how closely they fit the purpose. So is this impression of design kind of like, looking at the old man on the mountain or seeing faces in clouds or something? Is it kind of marginal but not really compelling? Well, again, not according to Richard Dawkins. According to Richard Dawkins, it is overpowering the impression of design. He says that life overwhelmingly impresses us with the appearance of design, as if by a master watchmaker, impresses us with the illusion of design and planning. So he thinks it certainly looks that way, but that's not really true, because we know, he thinks, that we have another explanation for it. And so many people, especially in sciences these days, think that there is a second explanation for design other than arrangement by a mind, purposeful arrangement, and that is Darwinian processes. So our third point is going to look at those that show that there are structural obstacles to Darwinian evolution. There are physical reasons why it cannot do what is claimed for it. So in order to do that, let's just do a little refresher course on the theory of evolution. And of course, the evolutionary theory was first proposed by Charles Darwin, at least this particular kind of evolutionary theory. And Charles Darwin was thoroughly a man of the 19th century. And the 19th century was an age of great exploration. And Darwin participated in that. When he was relatively young, in his 20s, he set off on an exploring mission on the boat, the HMS Beagle, and he was the chief naturalist. And they sailed all around the world, entering many different ports and lands that were not very well explored up until then. And during his trip, he saw a lot of plants and animals that nobody had seen in Europe. For example, in the Galapagos Islands, which are on the equator about a few hundred miles west of Ecuador, he saw that there were a bunch of birds there, and when they were later analyzed, it turns out they were all finch species. And yet the mainland only had one or two finch species, and it seemed that a number of islands in the Galapagos archipelago had their own. And he said to himself, what the heck? How can each island in this archipelago have its own species of finch that exists nowhere else in the world? And he kind of scratched his head for a long time, but after decades of thinking about it, he came up with his idea of evolution by natural selection. He said, well, heck, suppose that there was a finch species that came to the island long, long time ago. Well, we know that the individuals of a species can vary. Some can be bigger, some can be faster, some can be brighter in color. And he reasoned that if one of those variations favored the survival of its possessor, then it would have a better chance of surviving and leaving offspring. And if that trait could be inherited, then more of the next generation will have that trait that helped it survive initially. And over the generations, that would spread and essentially fix or be possessed by all of the organisms in the species. And then perhaps another variation could come along and be favored, and another one, and over long times, maybe much change could occur. And that was actually a great idea. It was really good, and you could see where it might help explain those finch species. But Darwin took that idea and he ran with it. He said, wait, it not only can explain the variation in the finches on the Galapagos Islands, but maybe it explains the variation in finches all over the world. And if it explains variation in finches all over the world, maybe it explains how different kinds of birds came from each other, and maybe all different kinds of organisms and plants. And so he proposed it as a general theory to explain essentially the structure of all of life. So that is that grand scheme, not the particular scheme where it explains variation in finch beaks and so on, is what we're going to be concentrating on. And in order to analyze whether we think it does the job that people want it to do, we have to look closely at what's called Darwin's theory. Because Darwin's theory actually has a number of different parts to it, a number of different ideas which are all mushed together and called in the singular Darwin's theory. And three of the most important ones are shown here. There's in his theory, there's the idea of common descent, and that is that organisms which have some trait in common, maybe they can fly or something, that they inherited that from a distant ancestor. Maybe an ancestor in the past could fly, and maybe it led to this organism that could fly and that organism that can fly. And that's an interesting idea, but in my opinion, it's trivial because it doesn't say where the ancestor came from. And it doesn't say how those different branches came to differ from each other. It just says it was there in the beginning, this trait, and now different branches have it, and they got it by inheritance. Well, okay, but you haven't really explained much of anything. The second component of Darwin's theory is the idea of natural selection. That is that if a trait helps an organism to survive, then it will have a better chance at surviving and leaving offspring. And again, that's an interesting idea, but again, too, it's kind of trivial. Because who's going to deny that if an organism has an advantage, then it won't do better than an organism that doesn't? 99.9% of the scientific, philosophical importance of Darwin's theory is wrapped into the third component of it, and that is the idea of random mutation. That is, that the changes are powered by chance, by nothing, by no particular cause. They just occur by serendipity, and that serendipity could supply the fodder for natural selection to build over time the most intricate biological structures. So the critical claim of Darwinism is the sufficiency of random mutation. So that's what we're going to look at here. And it's not just me that thinks that that's the critical claim. Here's a fellow named Douglas Fatuma, who was president of an evolution society for a while, and he's written that the reason that natural selection acting on random mutation is important is that it's the central idea that explains design in nature. Before Darwin, the adaptations and exquisite complexity of organisms were ascribed to creation by an omnipotent, beneficent designer, namely God. So this chance, this randomness, is a substitute for a mind, for the creative mind of God. And so this, as I said, is the key to Darwin's theory, not the idea of common descent, not the idea of natural selection. Well, okay, Darwin proposed his theory, but he didn't demonstrate that it could do what he claimed for it. So let's ask ourselves, what has biology shown, and does it look like it can be explained by Darwin's theory? Back in Darwin's day, biology was primitive compared to what it is today. For example, molecules were theoretical entities. Nobody was sure if they really existed or not. Cells, which we now know to be the basis of life, were thought to be little pieces of jelly that people called protoplasm, not thought to be very much of a much, and thought that they could maybe spontaneously pop up on the seafloor. Well, of course, science has advanced quite a bit since then, and especially in the past 75 years. You should realize that before the year 1950, nobody knew what was the basis of inheritance. DNA was thought to be a structural molecule. It wasn't until around 1950 that DNA was shown to carry the information of heredity. And in the early 1950s, Watson and Crick discovered the shape of DNA, the double helix. In the 50s, the shape of proteins were discovered, showing that they were the machinery of life. In the 60s, the genetic code showed how DNA could carry all that information. In the 70s, 80s, and 90s, different techniques, PCR, the polymerase chain reaction, cloning, sequencing, increased the capacity of biological research to go faster and faster. And the bottom line of all of that research, I think, the most important empirical conclusion is that the cell is run by machines. Literally, machines made out of molecules. This is the cover of a professional science journal called Cell. As you can see, it's a special issue on molecular machines. And if you look in the lower corner here, this looks like something like Paley's watch down at the bottom. If you turn the cover and you look at the table of contents, you'll see articles such as "The Cell as a Collection of Protein Machines," "Polymerases and the Ribosome, Machines Within Machines," "Mechanical Devices of the Spliceosome, Motors, Clocks, Springs, and Things." And this is 1998. You can say, well, you know, what have you done for me lately? And this has only gotten more and more and more sophisticated. Here's from the 2020s, where the structure of something called ATP synthase was elucidated. ATP synthase is actually a turbine engine which uses a flow of acid from outside the cell to inside the cell to synthesize the energy-charged molecule, ATP, that keeps your body running. So the question is, can Darwin's theory explain what biology has discovered about life? Not protoplasm, not jelly, but the machinery now of life. And in the past 30 years, I've written 3 main books: Darwin's Black Box, The Edge of Evolution, and Darwin Evolves, in which all 3 of them I used to argue that Darwin's mechanism is grossly inadequate to explain the molecular structures of life, and that a firm conclusion of purposeful design is empirically justified. That is, it's justified by the evidence. So let's start with this first one. I'm going to try to go through this one in the first part, and we'll talk about some of the other work in the second part of my presentations. Okay, in Darwin's Black Box, I concentrate on an observation that Darwin himself made in The Origin of Species. In The Origin of Species, he gives his proposal, but then he has a section called Organs of Extreme Perfection and Complication, where he talks about things like the eye, which are really, even then were known to be incredibly sophisticated. And he kind of waves his hands and says, well, start with this and add this and do that. And then he says, why, look, it's not a problem at all. But then he said, if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down, adding, I can find out no such case. And in this, Darwin was insisting that his idea of evolution had to work slowly, in tiny steps, over long periods of time, because if things appeared to improve quickly in large leaps, then it would look compellingly like something other than chance was involved. So Darwin always insisted on this gradual, slow evolution. So let's ask ourselves, well, what what sort of an organ couldn't or doesn't look like it could be produced slowly? And we can't, this is a rhetorical ploy to say could not possibly, because science can't prove a negative. But we can say, what sure looks like it can't be formed by numerous successive slight modifications, and for which there is no evidence that it can. Well, I wrote in Darwin's Black Box that a system that resists Darwin's criterion is one which is irreducibly complex, or has the property of irreducible complexity. And that's a fancy phrase, but it stands for a simple idea. It just means that you've got a system that has a number of different parts that interact with each other, and by interacting with each other, they produce an effect that none of the parts by itself could produce. And again, it's good to see an example. So here's an example from our everyday world of that idea. And this is just a simple mechanical mousetrap. You buy it at a grocery store or somewhere. And a mechanical mousetrap can have something like a big wooden platform and a spring, and the end of the spring has to have a part that pushes against another part called the hammer, which is the part that squashes the mouse, and there's a holding bar that has to keep the hammer back in place, and there's a catch in which the end of the holding bar has to extend. Now the trap needs all these parts. If you're missing the hammer or the catch or the base, it's not like the trap works half as well as it used to, or a quarter as well as it used to. It's broken. It doesn't work at all. So this is the idea of irreducible complexity. And the problem it presents to Darwin's theory is that if you have to work by numerous successive slight modifications, improving a function along the way, how are you going to make something like this? Would you start with, say, the platform and hope to catch mice inefficiently? Maybe when they ran over it, they'd trip and fall and hit their head or something. And then maybe you could add the holding bar here, this holding bar, and then maybe if they fell, they'd impale themselves on the holding bar, and that would be, nah, can't do it like that. So irreducibly complex systems strongly resist Darwin's requirement of numerous successive slight modifications. So the question then becomes, are there any irreducibly complex biological systems? Or especially now that we've reached the foundation of life, the molecular and cellular foundation of life, are there any such structures in the cell? And the answer is, yeah, they're all over the place. You can barely find one that isn't. Here's a great example. Here's something called the bacterial flagellum, which is quite literally an outboard motor that bacteria use to swim. And here's the propeller part. This actually rotates around and around and around, pushing against the water and pushing the bacterium forward. It's attached to the drive shaft by something called a hook, or hook region, which acts as a universal joint. The drive shaft is attached to the motor, which rotates around and around. There's a stator that keeps it in place. There's bushing material to allow the thing without leakage from the cell. If you take away the hook, if you take away the drive shaft, if you take away the motor, you don't have a flagellum that spins half as fast as it used to, or a quarter as fast. You've got a broken flagellum. So much like the mousetrap, this is irreducibly complex. Now, I'd like to show audiences this drawing because they quickly apprehend that this is a machine. It's not like a machine, it's not analogous to a machine. It is a real machine made out of molecules. And again, if you go back to the dictionary and you ask yourself, what's the definition of a machine? You find that a machine is a complex device consisting of a number of interrelated parts, each having a definite function to perform a certain kind of work. Essentially, it's the definition of irreducible complexity. And you remember that the cell is filled with machines. So it turns out that Darwin's idea of numerous successive flight modifications is exactly the wrong kind of theory to explain what science has discovered in the cell. And I was just going to show you some details of the flagellum, but I'm not going to do it because of time restriction. Ooh, look at that. Ooh, don't you wish you knew what that was? Sorry, I'll tell you about it at break if we have time. Now, it turns out that irreducible complexity is a two-edged sword. It has twofold importance. Number one, it's an obstacle Darwinism, as we just mentioned, and it also is evidence of purposeful design because you have a number of parts arranged for a function. So this is both evidence against Darwinism and positive evidence for design. Okay, so that was the point that there are structural obstacles to Darwinian evolution, irreducible complexity. But again, we are confronted every time we see biology discussed with claims that science already knows that Darwinian processes do explain this. So I want to briefly show you that those grand Darwinian claims rest on undisciplined imagination. And to do that, I'm going to rely on this book here. This was called The Way of the Cell. It was written by a man named Franklin Harold, who was a very prominent microbiologist at Colorado State University. He died about 20 or so years ago. And he was writing about his ideas for how cells work and how they arose. This was about 5 years after Darwin's Black Box was published, and he paused for a moment in his book to write about the idea of intelligent design. And he wrote the following. He said, "We should reject, as a matter of principle, not a matter of evidence, as a matter of principle, the substitution of intelligent design for the dialogue of chance and necessity." And he refers to my book. "But we must concede that there are presently no detailed Darwinian accounts of the evolution of any biochemical system, only a variety of wishful speculations." Well, okay. So he makes 2 points here. Let's pause a second to notice what the 2 points are. The second point, and let's go in reverse order. The second point is that he says there are no explanations, only a variety of wishful speculations. Wishful speculations, kind of like the Just So stories that Rudyard Kipling wrote for children at the beginning of the 20th century. I'm sure you know them, how the elephant got its trunk, how the rhinoceros got its horn, how the bacterium got its flagellum. You know those stories, right? It's kind of funny, but if you think about it, it's astonishing that a theory that is claimed so much, to explain so much, is a little better than a children's story. The second point, the first point, but let's take this now. He said we should reject intelligent design as a matter of principle. What principle? What principle is that? It turns out he doesn't say. He just writes what I showed you there and then goes on to talk about other things. Nonetheless, I have a good idea what the principle is that he's thinking about, and it's seen on the next slide here. And that is that intelligent design seems to point strongly beyond nature. It seems to have philosophical, maybe even theological implications, and that makes a lot of people nervous. And some people say science should steer well away from theories that have such extra scientific implications. And I understand what they're saying, but I disagree. When I was studying science, when I was being trained as a scientist, we were told that science is supposed to follow the evidence, wherever it leads. Let other people worry about philosophical or theological implications. We're supposed to be true to our evidence. And I thought so then, and I think it continues to be relevant today. Well, that was in 2002, but the lack of explanations still continues. Here's a paper from 2022 saying that modern evolutionary theory has little to say about the actual history of life and nothing about the origin of life. People say this all the time when they're proposing a new idea about how we might try to explain where life came from. And if you look in the literature, you'll find lots of admissions that we haven't a clue, but people circle the wagons when an outsider starts to question the consensus. And it's not just big things that are difficult to explain. Even the very smallest features of life that require arrangement, purposeful arrangement, putting together. Here's something called a disulfide bond. You don't have to worry about what that means. It's just when two parts of a protein kind of come up and link together, kind of like a hook and eye latch, like this. Okay, a hook and an eye latch, you need the two components. It's not going to hold a door shut if you just have the eye. It's not going to hold the door shut if you just have the hook. You need them both. Same thing, for a disulfide bond, you need not just one cysteine, you need two cysteines to be able to form the disulfide bond. About 15 or how long ago, a little more than 15 years ago, somebody proposed a way that you might explain a disulfide bond. And this fellow here, who's a prominent biochemist, Eugene Koonin, who was the editor of the journal to which a paper was submitted, was ecstatic that somebody might be able to explain this. He said, the idea of this paper is as brilliant as it is pretty obvious in retrospect. A novel solution is offered to the old enigma of the evolution of complex features in proteins that require two or more mutations. Emergence of a disulfide bond is a straightforward example. Let me emphasize, this is the easiest, the most minimum cooperative structure that exists in biology. And people, it was an old enigma. Did anybody tell you it was an old enigma back before this paper was published? No, nobody admits to any shortcomings until somebody thinks they've gotten a solution. Let me just say, in the 18 years since this paper was published, nobody's heard from it again. The idea had many, many problems to it. And the enthusiasm that Eugene Koonin expressed here has more to do with the seriousness of the problem than it has to do with whether or not this proposed solution actually worked. So again, I want to emphasize here, Darwinism, we are always told its claim to explain cells, the genetic code, molecular machines, genetic networks, phyla, literature, music, politics, the law, love, the universe, and even mind itself. You can read lots of articles where it explains these things. But it has a little bit of trouble with a disulfide bond. So it's like somebody saying, yeah, we can explain this supercomputer. You know, I got this, no problem. And it's being held in a shed which has a hook and eye latch on the outside. Well, I can't really explain that on the outside. You know, that's bluster. That's essentially braggadocio. It can't explain any of these things. There's no reason to believe that. So I want to end with this image that, in fact, Darwin's theory of evolution is a Potemkin village. It was established in the 19th century when little was known about biology, and now biology, the study of biology, has increased by leaps and bounds, and Darwin's theory was left in the dust. The front part of the village is the resemblances that creatures have to each other. It's clear that some features of different sorts of organisms resemble each other. The missing building behind is how in the world could such fantastic things happen? How can we explain such things? So that was the point that the grand Darwinian claims rest on undisciplined imagination. And so let's go to the conclusion that right now we have extremely strong evidence for design and no evidence that Darwinism can do what it claims to do. You might be saying to yourself, okay, well, yeah, I see that there's no evidence for design or for Darwinism, but remind me again, what is that strong evidence for design? And it's exactly what Richard Dawkins said, that life overwhelmingly impresses us with the appearance of design, as if by a master watchmaker. That is the evidence for design, the purposeful arrangement of parts. And we can see it right in front of our eyes. So the point is that we have in hand strong evidence and just bluster that anything else besides the mind can explain life. So in this first part, I wanted to let you know that a conclusion of intelligent design is rationally compelling. It's compelling based on the physical evidence and based on ordinary logic, inductive logic that science uses all the time. So thanks very much for your attention.
Okay, thank you, Dr. Behe. We're going to go ahead and take a short break and stretch your legs a little bit, and we'll be back in 10 or 15 minutes or so, and we'll move into part 2, and we'll have some interesting things there. So thank you again, once again, for coming.
So interesting. It's absolutely amazing.
Yeah, it's really
You
crazy.
explain it so well.
Yeah, it's pretty straightforward.
Yeah.
All right, somebody should turn me off here.
Yeah.
Let's see, here we go.
Okay, we'd like to get started again, if everybody can come to their seats. It's really hard to compress into a few minutes what could take days and days to talk about, but here we are. I would like to encourage you all, if you wish, to sign up in the back. We were collecting email addresses if you'd be interested in meetings similar to this in the future. So if you're at all interested, please sign up in the back. We'd really appreciate that. As we start part two, I'm going to introduce you to a movie that's going to appear 2 weeks from today in theaters all across America. Here, it's called The Story of Everything, and it's going to be in theaters across America. So what I'm going to do is show you just a 2-minute clip, and then we'll get into the next part of this message. So go ahead, Roger.
Today, I'm going to tell you a story which may seem very strange.
Galileo, Kepler, Newton.
Each tried to explain events in the history of the universe.
Has the universe always been here, or is it finite?
Is there something else that would lay these questions to rest?
It reopens that question of ultimate meaning.
How in the world did this start?
The simulation theory?
The multiverse. You can't trust what's in front of your eyes.
Come on, that's ridiculous. That belongs in the movies.
We want to take our metaphysical hypotheses and see what they point to.
And I can remember him saying,
Here is evidence for what can only be described as a supernatural event.
he himself made a discovery that shook his personal philosophy.
The fact the universe sprang into being at a definite moment seems to me theological.
And it is science that has revealed this.
We're dealing with a system of manifold, complex design.
The force compared with the weak nuclear.
1 part in 10 to the 10th.
Not too fast, not too slow. Turned out to be the tip of the iceberg.
We associate information with a rational intelligence behind it.
It had an uncanny resemblance to a digital bit string, very much like an information carrier.
You can read the same segment forward to get one protein and backwards to get another.
It struck us with a tremendous impact.
Without guidance, we would get a life-unfriendly universe. Many organisms have beauty beyond anything that's relevant for their survival value.
The concept of life as a cosmic phenomenon should have many consequences. The question then was, what does one do about it?
Okay, the movie premieres in 2 weeks from today, and it's only going to be playing for approximately 1 week in a few local theaters. So if you're really interested in it, they have the next slide up here. It's going to be showing in Trexlertown and Center Valley in approximately 2 weeks from now. If you want more information, you can see me or look it up. It's called The Story of Everything. And on Saturday, May 2nd, the group of us are going to go to the 3 p.m. showing in Trexlertown, which would be exactly 2 weeks from today. So if you're interested in seeing that, take a look. Professor Behe? Okay, you're on. Thank you.
The trailer for is really terrific. It's put out in part by Discovery Institute, of which I'm a member. Yes, I have a little bit part in it too. I do get a few royalties from it, but that's not the reason that I'm suggesting. No, just kidding. No, it's great. It talks about not only biological design as I'm doing here, but design based on the structure of the universe. The universe had a beginning, which was unexpected and fine-tuned, and life requires design to start and to continue, and it talks about the information in life. So I really urge everybody to go see it if you've got the time. Okay, so we talked about the problem of irreducible complexity and the purposeful arrangement of parts, and I told you that right now we've got strong evidence for design and little evidence that Darwinism can do the grand things that people have claimed for it. But what can it do? It can do some things. There's antibiotic resistance. There's no reason to think that that can't just happen spontaneously. And things like sickle cell disease, that's can be ascribed to Darwinism too. And so in the past 30 years, after I wrote Darwin's Black Box, science has advanced by leaps and bounds. It's really crazy how much it has continued. And in 2007, I wrote a second book called The Edge of Evolution, which asked the question, what's it reasonable to think that Darwinism could do, given how fast mutations occur and other things? And the short answer is not much. And then as more and more results came in, the question of what does Darwinism do could be addressed. And the answer to that is really astounding, and that's going to be the topic of this second part, and it's also the topic of my most recent book in 2019 called Darwin Devolves: The New Science About DNA. And it's interesting. Let me start with a few quotes from folks. This fellow right here, named Ronald Fisher, back in 1930, wrote a book called The Genetical Theory of Natural Selection, in which he said that, well, if you've just got two different variations of 100 genes, you can have an astronomical number of possible combinations. So it seems that there is no limit to the number of combinations that one can have in your genetic information. In 1944, this fellow here, G.G. Simpson, a towering figure in evolutionary biology, wrote Tempo and Mode in Evolution, saying that why even small mutations that we see going on around us now show that evolution could go faster than even than that's shown in the fossil record of a horse. In other words, it could go at breakneck speeds. And these are part of what has come to be called the neo-Darwinian synthesis, or the modern evolutionary synthesis. These are part of the founding books, series of books, that have gelled together into neo-Darwinian theory, which is still the dominant theory today. But both of these were written before it was known what genes consisted of. Nobody knew what a gene was. You could say if a gene has two different components, then if we have 100 of them, you can have 2 to the 100th different combinations. But they didn't know what a gene was. It wasn't until the early 1950s that it was known that DNA was the genetic material, and that Watson and Crick discovered the structure of DNA. And it turns out that back then, people simply didn't have the means to follow evolutionary change and figure out what was going on. But as we know, science advances with new technology. Before the invention of the telescope, people knew something about the external heavens, but with the invention of the telescope, could learn much, much, much more. It turns out that just recently, within the past 25 or so years, maybe 35 years, new technology has been invented that allows us to probe what evolution actually does at the molecular level, because mutations are changes in molecules, in DNA, and the proteins that they code for. So in order to evaluate what evolution is doing, you have to track those changes in the DNA, see what they are, and how they change in the population over time. Couldn't do that. It was like studying the universe without a telescope. So a lesson is that forget about what you've learned about how evolution works. It's as irrelevant as what people thought about how the universe worked before the invention of the telescope. In the past 30 to 35 years, there has been a revolution in doing something called sequencing DNA. As I'm sure almost everybody knows, DNA is a polymer, has a number of units strung together, and it is that sequence of subunits, something called A, C, G, and T in DNA that carries genetic information from generation to generation, much as the sequence of letters in a book carries information to people who read it. In the past 35 years, it was impossible to do before, but in the past 35 years, fast and cheap and efficient ways have been invented to sequence the entire genomes of whole organisms. And just to a reminder, the DNA content of the genome of humans is 3 billion nucleotides, 3 billion units. One these days could sequence somebody's genome in an afternoon. Before 30 years ago, you couldn't do it at all. And bacteria and viruses and other things have smaller genomes. People can, they're assigned to, grade school students to do if you've got the equipment in your classroom. So you can now follow the changes in DNA as evolution occurs. Another thing, though, that evolution needs is a lot of organisms and a lot of generations, because if one organism gets an advantage, then it's got to have time to compete and leave more offspring and take over the population and so on. And heck, if you do that with, say, cows or horses, you're not going to be able to have enough organisms and enough generations. The average research grant lasts 3 to 5 years. You can't do that in 3 to 5 years. But if you do it with bacteria, the numbers and the generations become feasible. And that's what was started by this man here, Richard Lenski, who's a microbiologist at Michigan State University. In the early 1990s, he started to grow a bacterium called Escherichia coli, E. coli, in his lab. What he would do is take a small 10 milliliter aliquot of broth, nutrient broth, seed it with E. coli, let it grow overnight, and it would grow 6 or 7 generations, just because bacteria are so small they reproduce quickly, and because they're so tiny, even 10 milliliters or a third of an ounce or so can have billions and billions of cells in it. And the next morning he came in and he took a little drop of that and he used that to put into a fresh 10 milliliters of medium. And he let that go 6 or 7 generations overnight. Next morning he came in and did it again. Next did it again, and then did it again. And currently they're up to 75,000 generations of E. coli born and died in his laboratory. Trillions upon trillions of cells. So they have the numbers and the statistics to see what is really going on. How is it changing? He did this specifically because he wanted to see how the E. coli would evolve. And he started in the early 90s, and sequencing then was starting to get started, but it had not advanced very far. So what he saw in the early days is that after a while his bacteria could change. Some of them would start to grow faster. And he'd write a paper about that. And some of them would get bigger and they would out-reproduce other ones. And he'd write a paper about that. But he didn't know what the changes in the DNA were that were causing these phenotypic changes in shape and so on. But then, about the year 2000, they were able to track down one change in a bacterium that gave it a pretty big advantage, kind of serendipitously. I won't go into it. And they published about that. And he says that 12 population, they did it in 12 different test tubes just for replication's sake, 12 different ones. And all 12 strains of E. coli lost the ability to make ribose, which is a kind of sugar. And he said these ones that lost ribose function, ribose minus, were about 1 to 2% more fit than the progenitor. Now, 1 to 2% doesn't sound like much of much. That means that the ones with a mutation leave 101 cells in the next generation for every 100 that the ones that don't have the mutation leave. But over 50 generations or 100 generations, they take over the whole colony, and that's 2 weeks in the lab. So this is actually a huge advantage in these circumstances. So the point is that a beneficial mutation involved the loss of a gene that was already there. And he was only able to do this in the year 2001 because new techniques had been invented. A few years later, he and his students and postdocs and stuff, tracked down a number of other mutations, a handful of other mutations that also helped the bacterium grow better and outcompete other non-mutant bacteria in his laboratory. And these are little graphs of the different genes, pyruvate kinase F, nicotinamide adenine dinucleotide are the names of several genes. And it turns out that they have multiple different mutations in the 12 different strains that he was growing for duplication state. And the reason that mutations occur at different places in the gene is because genes can consist of thousands of different nucleotides. And if you hit it here, or you hit it there, or a number of places, you can break it. You could break it at a number of different places. Just like you can break your computer by hitting it here, or hitting it there, or hitting it here. Around the year 2010, they were still working, and I was reading their very good work, their fascinating work, and also a number of other folks who were doing relatively similar stuff, although Lenski's lab was the preeminent one. And I wrote a paper called Experimental Evolution, Loss of Function Mutations, and the First Rule of Adaptive Evolution. And in it, I said that we should recognize what I called the first rule of adaptive evolution, the first thing that we should expect evolution to do, and that is to break or blunt any functional coded element, and that's just technical language for saying a gene or a regulatory region that turns on or turns off a gene. Break or blunt any gene whose loss would yield a net fitness gain. That's more technical talk for saying leave more offspring. So break anything, and if you break it, if it leaves more offspring, that's the first thing you should do during evolution. And people didn't like this. I got a lot of love letters because of it. But in the year 2016, let me just say, why should we expect such a thing to be true, this first rule of evolution? Simply because genes are machines. Genes code for proteins, and proteins are parts of machines. And so in order to improve a protein, there might be one or two places where you can tweak it, just like your computer. You might be able to tweak it here or tweak it carefully there or there, and that might help. But you can break it in a lot of different places. It's real easy to come up with a mutation that would break something, a lot easier than to improve something. And if there's an advantage to be had by getting rid of something in some circumstances, evolution will find that much, much faster than it would a constructive mutation. And again, in the year 2016, Lenski's lab published the definitive results of their experiment, which at that point was up to 50,000 generations. They published it in the most prestigious journal in the world, Nature, Tempo and Mode of Genome Evolution. And they put out two tables of genes that had acquired mutations that gave the biggest advantages for the E. coli to survive in their lab. And they consisted of 30 different genes. And of those 30 genes, approximately 30 were ones that had been degraded or outright broken. So it was following the first rule of evolution. Since then, other labs have done similar things and gotten pretty much identical results. Here's a paper from a different lab by a prominent scientist named Bernhard Paulsen, looking at aggregating the results of a bunch of different work on E. coli, saying that if you look in nature, the mutations in nature are very mild and unlikely to affect protein function. But of the 33,000 mutations acquired in laboratory evolution experiments, they result in more severe amino acid substitutions that are rarely achieved by natural selection. That's a kind of a circuitous way of saying these things all break the proteins, the genes in which they occur. And these are the ones that are being selected positively in the laboratory under various conditions. Here's just more quotations saying the same thing. The mutations acquired through laboratory evolution are more severe and are rarely found in nature. Laboratory and natural selection pressures are largely disjoint. But in the laboratory, we're just seeing how they simply grow and follow Darwinian processes. We're not seeing what's going on in nature. So we see that when Darwinian processes are allowed their full reign, then they break stuff. People have looked at other microbial organisms too. Saccharomyces cerevisiae, which is yeast, baker's yeast. Mycoplasma mycoides, which is one of the smallest kinds of cells. And they do exactly the same thing. In this 10,000 generation experiment on yeast, of the top, 50-ish, 50 again were ones that broke genes. For the mycoplasma, and the mycoplasma has the least amount of DNA, a very small amount of DNA as these things go. And nonetheless, it still improved its growth and outcompeted its ancestor by breaking or degrading stuff that it already had. How does that work? How do you get better by breaking something? Well, suppose you had this sweet ride right here. Hey, I love this. And somebody comes up to you and draws a gun and points it at you and says, unless your car gets better gas mileage by tomorrow, I'm going to shoot you. What's the fastest, easiest thing you could do to get better gas mileage? Pardon? Well, more than Priuses are designed. If you had to work with your old car, what you could do is throw away a door, throw away a hood, lighten it, and that would help it get better gas mileage. Of course, it doesn't, explain where doors or hoods or such things come from, let alone where a car comes from. But if you had to improve your gas mileage in a short time, this is the way to go. So here's an image I want to leave you with of Darwinian evolution at work. I got this picture off the internet, so you know it's true. And the story behind this is supposedly that these are army soldiers in some Central South American country, I think, and they got a tip that up the road here there was a gang growing plants for drugs and a drug refining operation. And so they were going to raid the compound, but the drug makers got tipped that they were on their way, and there was only one road leading to the compound, and they blew up the bridge leading to the compound. Now, from the point of view of the drug makers, this is a beneficial mutation. They will live another day. Maybe they'll be able to scatter and regroup somewhere else. This is the kind of thing that Darwinian evolution does. It can blow up bridges and in that way improve an organism's chances. Sometimes it literally does that. For example, many people live in areas of the world where malaria is a deadly threat. And malarial parasite is a single-celled creature that gets injected by an infected mosquito, and it can travel around and attach to a red blood cell and enter the red blood cell and eat the hemoglobin inside and trash it and use that energy and materials to make more malarial cells. But some people in West Africa have a mutation where the protein that the malarial parasite binds to is thrown away. They have a mutation breaking the gene that usually makes that. Now that protein usually does something helpful, but it's better for them to lose that protein and not be invaded by malarial cells than to keep the protein itself. So this is what we now see happens. Now, due to the DNA sequencing revolution, we now know that the following. We had known, the old information was that of those mutations that affect an organism, about 99% are detrimental. They hurt the organism. But nobody cared about that because if the organism got hurt, it'd just die and it wouldn't leave genes for the future, and that's how natural selection works. But the new information is that of even beneficial mutations, the great majority break genes or degrade function. And these beneficial degradative mutations are a whole different kettle of fish because they are selected by natural selection. They increase in the population. And once they fix, that gene is gone forever. So it turns out that Darwinian evolution is actually devolutionary. It breaks things it breaks things much more easily than it makes them. I'm going to skip this for purposes of time. So yes, here we go. Darwin's mechanism is dominated by what I call poison pill mutations. That is, positively selected, helpful, but loss of function mutations. And it works primarily by squandering genetic information it already has for short-term gain. And we biochemists, love E. coli and Saccharomyces and stuff like that, but I know other people are more interested in bunny rabbits and birds and larger organisms. Turns out it works the same with larger organisms too. For example, dog breeds are one example that Darwinists have pointed to for a long time. They say, look at the great variety of dog breeds that we, human breeders, have been able to produce in only a few hundred years. Look at all that variation that's available for natural selection. This shows how Darwinian evolution can work. And they were more right than they knew. It does show how Darwinian evolution works. But when we look at the DNA of these dogs, we see it works by devolutionary mechanisms, not by constructive ones. Turns out that DNA sequencing is now so cheap and easy that some scientists who are also dog lovers says, let's sequence the genomes of every pure breed of dog that we can get our hands on to see what genes cause the characteristic traits of many of those dogs. And we won't go through it, but the long and the short is that overwhelmingly they are degradative mutations. If you want a dog with curly hair, you break a certain kind of keratin that the dog makes. If you want a dog that's short, like a dachshund, you can break components involved in the growth of its leg. If you want a dog that's really highly muscled, you can break a gene called myostatin, which tells the body when to stop making protein. You can produce any trait, whether it looks impressive to you or not, by breaking something rather than making something new. Another example are polar bears. It turns out that polar bears branched off from brown bears, grizzly bears, maybe a couple 100,000 years ago. And again, because of this impressive ability to sequence DNA, people sequenced the DNA of brown bears and polar bears specifically to ask the question, what causes a bear to turn into a polar bear when it started off as a brown bear? And here is the first paper that reported on it. And they said, we assessed this specific substitutions by computational predictions, essentially by computer. They estimated it because you can't breed polar bears as well as you can E. coli. And they wrote that a large proportion of about 50% of mutations were predicted to be functionally damaging. And this is actually an underestimate because a couple of genes have multiple mutations in them, and at least one of them is damaging, while maybe another one is not estimated to be one. So the long and the short is that you can make a polar bear from a brown bear, but it's by devolution, not by evolution. And it's interesting, too, that people are looking at larger structures in biology. A fellow named Eugene Koonin, we heard from him before, we're looking at yeast in various branches of yeast. They're looking at the yeast sequences. Yeast, it turns out, has a whole wide range. It's like saying animals. Saying yeast is like saying different animals. There are all sorts of different types of yeast. And he's looked at ones that were thought to be earlier in the evolutionary timeline and some later and so on. And they found that the evolution is mostly reductive. He says these and many other cases of reductive evolution, means they get rid of things that their ancestors had, are consistent with a general model composed of two distinct evolutionary phases. There is a short, explosive innovation phase that leads to an abrupt increase in genome complexity, followed by a much longer reductive phase. Quantitatively, the evolution of genomes appears to be dominated by reduction and simplification, punctuated by episodes of complexification. We can put that differently. We can say that periodically large amounts of information are injected into a biological category, and then it diversifies mostly by losing some of that information that was injected into it. Okay, how can we view this thing? Well, let's take a look at a number of different kinds of organisms that have that were very explosive, that produced a number of species. For example, the Galapagos finches. We know that there are about over a dozen different species of finch on the Galapagos islands, but they are supposed to be related to finches on the mainland. So how are they, let's look at the classification of those finches versus their ancestor. And here's the different levels of biological classification. I'm sure you remember these from high school: domain, kingdom, phylum, class, order, and so on. Both the ancestor and the descendant of these finches were eukaryotes, which means organisms that have cells with nuclei in them. And both the ancestor and the descendant are animals. Ancestor and descendant are both animals that have backbones, chordates. They're both birds. They're both songbirds. They're both in the family Thropidae, and they differ in genus and species. Those 12 species of finches were categorized into 4 different genera. Well, let's look at this. Each level here is kind of like an order of magnitude in difference. You know, saying that something is an animal is, only, a small fraction of eukaryotes are animals, and small fraction of the groupings of animals are chordates, and so on. So we can think of these different levels as kind of like different orders of magnitude, or put it differently, as different places in a number that has, place values like 1 million or so, millions and hundreds of thousands and so on. So let's compare the ancestor and descendant of the Galapagos finches. And to make it more interesting, let's think of it as quantities of money, a dollar value. Oh yes, I'm sorry. So here we see that they are the same. They're the same all the way down to the level of family. They differ only in genus and species, the 2 lowest categories of biological classification. If we did that with our sum of money, then that means that over the 2 million years that the Galapagos finches are thought to have diverged from mainland, it's like the difference between $213,754.36 and $213,754.83. They differ just in the pennies and dimes columns. If you had put your money in, the Bank of America, you would have a whole lot more change than that. If you do your taxes, you know that the IRS tells you just to round off to the nearest dollar. Ignore the pennies and dimes columns. If we follow that advice here, we would ignore the difference in all of those finch species. Another very abundant species-rich organism are cichlid fish. Turns out there's a lot of cichlid fish in what are called the Great Lakes regions of Africa, Lake Tanganyika, Lake Malawi, and another one called Lake Victoria. And Lake Victoria is particularly interesting because it's kind of shallow, and it actually dried up about 15,000 years ago. And everything in it was killed off, of course. But then it refilled after a couple thousand more years. Some rains came back. And cichlids came in. And there is about 1,000 species of cichlids that exist in Lake Victoria, which exist nowhere else on the face of the earth. So they must have evolved right in Lake Victoria in the past 15,000 years. But if you look at all of those fish, it turns out that they are all eukaryotes, they're all animals, they're all chordates, they're all fish. They differ only in the last two categories of biological classification. They're all members of the cichlid family, and there's cichlids all over the world, in South America and many other places. A fellow named George Barlow wrote a book back in 2000, The Cichlid Fishes, Nature's Grand Experiment in Evolution, in which he marveled at this. He says, yeah, you get all of these things, but you know, all cichlids have the same characteristics, but they have some ephemeral differences. And he says, why, if you look at this, some taxonomists even want to do away with binomial nomenclature, getting rid of genera and species, because it seems so changeable and doesn't really get to the characteristics of the family. You can look at a bunch of other, what I call luxuriantly evolving groups. We looked at finches and cichlids, anoles, which are reptiles, honey creepers, which are birds, fruit flies and beetles, which are insects, silver swords and lobelias. And you look at those, they can explosively speciate. You get a bunch of species, fewer genera, and no families or higher categories. So how do we explain that? In my book, Darwin Devolves, I said, well, maybe we can explain it by calling it something called the family line, and realizing that there is such a thing as Darwinian evolution, and it's self-limiting. 3 factors restrict Darwinian evolution at the highest levels of biology by promoting it at the lowest levels: random mutation, natural selection, and irreducible complexity. And how does that work? The first rule of adaptive evolution allows organisms to quickly adapt to their environment by devolutionary processes. So if an organism goes into a new environment, say a grizzly bear goes into the Arctic, or a cichlid goes into Lake Victoria, or Galapagos go into an archipelago, they fiercely fight to survive. And if their beak gets a little larger, because the gene that would make it the right size has broken, and it allows it to grow larger, they can survive, eat larger seeds, maybe in times of drought. They can adapt by devolutionary processes, but once you break that gene for your advantage, it's gone. And now when the next change in the environment comes along, you're more restricted than you were before. So undirected evolutionary change rapidly bogs down before the family line. And so a prediction of this idea is that unique information inaccessible to unguided processes like Darwinian evolution, is required at the level of family and higher. In other words, yeah, evolution can tinker around the edges. It can maybe cause changes that we humans decide to classify as another species or another genus, but it doesn't reach what we classify as families. So that means that the limit of Darwinian evolution seems to be around the biological level of family. And just to kind of gauge what that means, it's like the difference between a dog and a cat. These are families in the mammalian order of Carnivora. That means if you have a dog, it will never give rise to a descendant that is in a different family, like the cat family or anything else. Same thing with the cat. It will not give rise to something in a different family. Okay, how am I doing for time, folks? I could go on for a little bit more. Why don't I go on for another 5-ish minutes? and then we can call it a day. Let me skip this. As you may know, not everybody agrees with this. There have been some criticisms of my ideas ever since I first wrote. And I had a few things that I was going to talk about, some scientific criticisms. But in interest of time, I'm going to skip this first one, which was a really, really good one. But anyway, skip. Oh, yeah, okay. Let's go back to this one. Okay, this is the book we've been talking about, Darwin Devolves. It was published in February of 2019. A little more than 2 weeks before it was published, the journal Science published a hit review of the book called The End of Evolution. Now, I was actually very pleased with this review because if you look at it, let me see if I can do this. Here, okay, here we go. I'm magnifying glass here. If you look at the authors of the review, first off, notice that there are 3 authors. I have never seen a book review that had 3 authors that it took to review the book. So I felt pretty good about that. Notice that the last author, if you can spot it, if I can hold my hand steady, is Richard Lenski, whose work I discussed. One of the chapters of the book pretty much concentrated on his work. So what did he have to say about it? Well, the review was relatively short, so I'm not going to say what he said in the review. I've written about all of this. You can see how I evaluate the review just by tracking it down on the web. But he also later on wrote about my book on his own blog, which he's called Taliamid Revisited, and I've never figured out what that means. But I'm going to look at this because he makes here what I think is his most important point, and he has as much space as he wants to talk about it. If you publish in a journal, they gave them 800 words to review the book. So he asks a swell question. He said, does Behe's first rule of adaptive evolution really show that evolutionary biology has a big problem? And his answer, of course, is no. Why does he say that? He says, because when it comes to the power of natural selection, what is most frequent, those devolutionary degradative mutations, versus most important, can be very different things. So some things can come along more frequently, but the big, changing things, they can be the important ones can be very different. And he goes on to write that Behe is right that mutations that break or blunt a gene can be adaptive. And he's right that when such mutations are adaptive, they are easy to come by. But Behe is wrong when he implies these facts present a problem for evolutionary biology, because his thesis confuses frequencies over the short run with lasting impacts over the long haul of evolution. Okay, so again, something important comes along that will have an unproportional effect. Okay, great. Tell me more about this, Richard Lenski, who is the expert, the world's leading expert on microbial and molecular evolution. Well, he presents 4 examples to illustrate his point. Or 3 examples. First, he says there's the cold virus, the rhinovirus, versus an Ebola virus. He says, well, if you catch a cold, you catch it very frequently, but it has some unpleasant effects, but you get over it. On the other hand, if you catch Ebola, few people catch an Ebola, but it has a really big effect. It can kill you. And the stock market, he says, well, yeah, he says, if you get 100 stocks, 95 of them might lose, but if 5 of them get a lot of money, then, you can be rich. So even if it's infrequent, it can do a lot of good for you. And something called Tiktaalik, which is the name for an organism they've found some fossils which they thought included bones which looked like an incipient wrist on a fish that was seemingly coming onto land. And he said that change, didn't happen very frequently, but it had a big effect. And those are his 3 examples. And I can say, yeah, but what do they have to do with what we were talking about? They don't address anything of what I just said. I talked about what went on in his laboratory, and he didn't refer to anything in his results that would have contradicted it. Okay, so some kind of viruses cause cold, some cause death. What does that have to do with anything? It doesn't even tell you how viruses evolve, let alone, how, they came about in the first place. What does gambling on the stock market have to do with the bacterial flagellum? Does that illustrate how the bacterial flagellum came about? Richard Lenski knows all about molecular biology. He knows all about the structure of proteins and molecular machines, and he has nothing to say. Tiktaalik. Okay, let's say that this organism was developing a wrist. What shows that it was by random mutation and natural selection? There's absolutely not. This is just an example of people pointing to similarities between this organism and that, and drawing untoward conclusions. Resemblances might be evidence for common descent. They say nothing at all about whether Darwin's mechanism is true. What's more, he writes, let me go back here, he says that what is most frequent versus most important can be very, very different things. This is a source of much confusion. When it comes to the power of natural selection, what is most frequent versus most important can be very different things. There is no such thing as importance in all of Darwinian theory. Here is Fisher's fundamental theorem of natural selection, which describes how fast a population will evolve based on how much variation it has. Here is the expected waiting time for a particular mutation to appear, given its mutation rate and so on. None of these factors gauges importance. This is the mutation rate, the waiting time, the rate of increase in fitness. None of them gauge importance. Frequency is there in both of them as the mutation rate. Darwinian evolution doesn't care about importance. It doesn't care if the next mutation that comes along completely stops development forever or not. It just comes along, that's what it does. It has no mind to plan ahead. So, okay, and the reason for that, I've shown in the slide before, is not a big deal. It's just that there's many different ways to break a gene versus improve one. And if there's a helpful effect that can be gotten by breaking a gene, then that's what evolution will take, because it has no way to see the future. So here's a slide. I'm not sure I'm going to go through this in detail. This just shows that the rate V of degradation is much greater than the mutation rate of construction, V sub con, by 100 to 1,000-fold. That means if you have to wait 100,000 years for a constructive mutation to come along, you would only have to wait 1,000 years or 100 years for a helpful degradative mutation to come along. That means if that were the case, that degradative mutation would have 99,000 to 99,900 years to spread in a population before the constructive mutation even showed up. This is a structural difficulty with evolution that can now only be realized because we now know the structure of DNA and we have been able to determine what evolution does at the molecular level because of the DNA sequencing revolution. Okay, and let me just read this one. These beneficial degradative mutations are ubiquitous. They are relentless and they are dominant. Once they get in the population, they will spread to completion and destroy the gene that is being selected. Okay, I have one other thing, but I'm going to skip that and say that. Yes, not everybody has agreed with me, and my responses to critics can be found in a number of places. So if you're interested to see what critics have written, you can look here as my website at Lehigh. Here I've got some stuff posted at the website of the Discovery Institute, and somebody set up a website for me. I don't know how to do that, but there is one, michaelbehe.com. It even has cool little videos and things. And also, if you really want to get into the nitty-gritty, I collected all of my responses to critics and put them together as a book. It's called A Mousetrap for Darwin, Me, I Answer My Critics. And this contains all of my responses. It doesn't contain the original criticisms themselves, but if you want to, you can track them down easily enough. Just go on any search engine, type in my name plus any common swear word, and you'll get lots of hits. Okay, and thanks again for your kind attention.
Dr. Behe, can I have you come back up here, please? Sure. And I'm going to ask the Share the Power board to come up here. And we just want to show, as the body of Christ in the Lehigh Valley who's gathered here today, we want to say thank you for sharing this with us. Share the Power board, would you guys come up here? And this event was put on by Share the Power in connection with Livingstone Fellowship. I'm the executive director of Share the Power and also the pastor of this church, so that's the tie-in. But we just want to take a moment as a form of saying thank you, we'd like to just pray blessing over Dr. Behe. So would you take a moment to bow your heads and let's pray for Dr. Behe. So Heavenly Father, we thank you for Dr. Behe and all the years that he has taken a stand for what is the truth. In the face of opposition, he has stood for the truth. And so we thank you for his life and his work in his field, which has been guided by you. Father, we thank you that you are a God who loves and you are a God who has designed. And so, Father, we walk out of here today encouraged, knowing that our faith is not wishy-washy, but our faith is solidly grounded in scientific truth, that there is a God who loves us, who designed this universe for us, and who has called us into fellowship with him. And so we speak blessing over Dr. Behe. We thank you for his life. We pray for protection, we pray for health, and we pray for many, many more years of wonderful service in his field and also for service for your kingdom. In Jesus' name, amen. And as a token of thank you, we'd like you to receive that. And I think we are done. Please see the table at the back. And if you have any questions, I don't know how long you're going to hang around, but come and talk to Dr. Behe. God